Welcome to part 2 of our lecture series on Chapter 3, Biology and Behavior. So, before we get started, I would like you to watch the concept video titled Neurons and Action Potentials in the folder titled Chapter 3, Biology and Behavior Lecture Videos. I will talk about this whole process once you come back from watching the video. Welcome back. So that video gave you an overview of the synaptic transmission process. And so we're going to explore that issue and you'll get a much better understanding of it. The function of every neuron is to transmit information throughout the nervous system. To do this, neurons communicate in two ways. Neurons talk to other neurons using chemicals, using neurotransmitters, and we talked a little bit about that in part 1. Neurons talk to themselves electrically, and we will talk about that in more detail. So now I would like you to watch the video 2-Minute Neuroscience Synaptic Transmission in the folder titled Chapter 3, Biology and Behavior Lecture Videos. And this is just gives you a look about what I'm about to talk through. And there is a naughty kitty behind me. Welcome back. I really like the 2-Minute Neuroscience series, and we are going to use more videos from that series throughout this chapter. So we're going to talk about, so far we've talked about the basic structure of individual neurons. And now we're going to look at how do two neurons communicate to each other. And this will help you understand why I needed you to understand the basic structures of an individual neuron. So now we're going to look at synaptic transmission. This is where neurons begin communicating with each other using neurotransmitters. So with synaptic transmission, one neuron releases chemicals into the synapse. And a synapse is a small gap between the neurons. So here we have what's called a synapse. This entire structure is called a synapse. We have the synaptic gap right in here. So our presynaptic neuron is the axon terminal from neuron A. Our postsynaptic neuron is the dendrites on neuron B. Now, our neuron A is called our presynaptic neuron because it's the one before the synapse, and our neuron B is called the postsynaptic neuron. So I want to look at the structures of the neuron in the synapse in more detail now. Let me check my next slide. So we're going to look at these individual structures and how this process of chemical transmission works. So now I would like you to watch the very short video of neurotransmitter synapse 3D animation. You'll find that in Chapter 3, Biology and Behavior Lecture Videos. Welcome back. So in that video, you get an idea of how quickly this process goes. And, you've got 100 billion neurons creating 100 trillion synapses. And so the video gave you a look at synaptic transmission, and then, action potentials, synaptic transmission, and then an idea of how the synaptic transmission is occurring throughout the brain with billions of billions of neurons at a time. Let me check my next slide. Okay, so back to our basic synapse. Here we have our presynaptic neuron, and this is our axon terminal. So the presynaptic neuron is neuron A, and we are looking here at one axon terminal. Within each axon terminal are what are called synaptic vesicles. And these are the structures that hold neurotransmitters. Now, each neuron is going to be programmed to use specific types of neurotransmitters, and I'll come back to that. So synaptic vesicles contain the neurotransmitters within the axon terminal, and once the neuron is told to engage in synaptic transmission, the synaptic vesicles, some of the synaptic vesicles, merge with the axon terminal membrane and just release neurotransmitters into this synaptic gap. Then you have this postsynaptic neuron, and so this is going to be a dendrite. Now remember, axons only send information. Dendrites only receive information. On our dendrite, we see what are called receptor sites. And it is receptor sites that either allow or block these newly released neurotransmitters into the postsynaptic neuron. Now, neurotransmitters bind with these receptor sites on the postsynaptic neuron, so the dendrite. And these receptor sites are protein molecules that allow neurotransmitters to flow into the postsynaptic neuron. So this is going to be the dendrite because only dendrites can receive neurotransmitters. And this works as a lock and key system. So as I just explained, each different neurons are programmed to use different types of neurotransmitters. So we're going to use the example of dopamine. Dopamine is our pleasure neurotransmitter. When we engage in something that feels good, we have dopamine being released, and we'll talk about that in more detail later. So here we have our presynaptic neuron, so the axon terminal, and we have the dendrite of the postsynaptic neuron. And on this dendrite, we see receptor sites. Now, receptor sites work as a lock and key system. So with this receptor site is programmed to receive norepinephrine and acetylcholine. It will not accept dopamine because this receptor site does not have a lock for dopamine to unlock. So this receptor site only accepts acetylcholine and norepinephrine. Now, every neurotransmitter type has a different molecular structure. So, why won't dopamine fit into this receptor site? Its molecular structure doesn't fit the lock. But the molecular structure of norepinephrine fits the receptor site. And acetylcholine will fit into this individual receptor site because the molecular structure of each of these neurotransmitters fit. Now, norepinephrine will not fit into an acetylcholine site, and acetylcholine will not fit into a norepinephrine receptor site lock. But this entire receptor site on the dendrite will accept norepinephrine and acetylcholine. Let me check my next slide. Okay. So here we have synaptic transmission. We have our axon terminal. We have our synaptic vesicles. And it is our synaptic vesicles that contain neurotransmitters. Once my presynaptic neuron is told to engage in synaptic transmission, some of the synaptic vesicles will merge with the axon terminal membrane. And then we see a release of neurotransmitters into the synaptic gap. My postsynaptic neuron, my dendrite, has receptor sites, and as long as the neurotransmitters released from our presynaptic neuron fit into the receptor site, this dendrite will welcome in that neurotransmitter. So we're using the example of dopamine. If my presynaptic neuron, my axon terminal, contains dopamine neurotransmitters, releases that dopamine, and my postsynaptic neuron receives dopamine, has receptor sites for which dopamine can unlock, then we have the full synaptic transmission process. If this axon terminal is releasing serotonin, but my receptor sites on my postsynaptic neuron only accept dopamine, this dendrite will ignore that serotonin. It won't allow any serotonin into the receptor sites. Now, we can also, let me check my next slide. One of the ways that drugs work, so I'm going to use selective serotonin reuptake inhibitors as my example. SSRIs. This is a category of medications that are used to treat depression and anxiety disorders, and also PTSD. Now, SSRIs are not the only type of antidepressant there is. We also have dual reuptake inhibitors, which work on serotonin and norepinephrine. There's multiple types of SSRIs and NSRIs. But an SSRI only works with serotonin. An NSRI works with norepinephrine and serotonin. So we're just going to look at an SSRI. So my presynaptic neuron is releasing serotonin. As long as my postsynaptic neuron will receive serotonin, it will accept that serotonin through its receptor site. Now, we can create drugs that mimic the molecular structure of neurotransmitters. Because again, every neurotransmitter, every type of neurotransmitter has a different molecular structure. The molecular structure of serotonin and the molecular structure of norepinephrine are different. So we can create drugs that mimic the molecular structure of whatever neurotransmitter it is that we're targeting. So here we've got norepinephrine, we have serotonin, and here we've created a drug that mimics the molecular structure of serotonin and tricks the lock, tricks the receptor site into accepting the medication and using it as a neurotransmitter. Amazing! I don't know how biologists, chemists, I don't know how they do this, but it's pretty incredible. So this is one of the ways that we can keep synaptic transmission going when someone is depressed. So when someone's depressed, the prefrontal cortex, that is the part of the brain that is flush with the forehead, the prefrontal cortex slows down. It becomes sluggish. And with drugs, we can alter how neurotransmitters are processed. We can alter how serotonin and norepinephrine are processed in the prefrontal cortex. Now, I'll come to more on that. So I want to talk about neurotransmitter reuptake. So we've discussed the basic process of synaptic transmission. My presynaptic neuron sends neurotransmitters into the synaptic gap to be received by the receptor sites of the dendrite on my postsynaptic neuron. When a neuron releases neurotransmitters, not all of the neurotransmitters can be taken in by receptor sites. So, let me just check. Okay. So when we look at our synaptic gap, here we see our receptor sites on our dendrite have received neurotransmitters. But there's still some leftover neurotransmitters in the synaptic gap. So it's not unusual for axon terminals to release more neurotransmitter than the dendrite can receive. And so once synaptic transmission has occurred and our postsynaptic dendrite has received its neurotransmitters, once the key has gone into the lock, the receptor site shuts down for a minute. And so we've got these leftover neurotransmitters, and the brain recycles them. So once the postsynaptic dendrites are full, once the key is in the lock, it can't accept any more of that neurotransmitter. So there's a recycling process called reuptake. And the axon terminal on our presynaptic neuron just takes those leftover neurotransmitters back. It's just a recycling process. I think of it like Thanksgiving. So, you know, the poor mom or dad responsible for doing all the cooking on Thanksgiving puts the food on the table. We've got the table here, which is the synaptic gap. The rest of us who did no cooking at all, been watching football all day, eat some of the food, but there's always leftovers. So the leftovers are put back in the refrigerator. And then, once we're hungry again, we pull out those neurotransmitters, we pull out the Thanksgiving food, and we eat it again. Or we can eat that, the leftovers. Let me check. So, when a neuron releases neurotransmitters, not all of the neurotransmitters can be taken in by the receptor sites. Because of this, neurons experience reuptake. And we just looked at the reuptake process. It's basically a recycling process. Now, the neurotransmitters' effects are halted by reuptake because now the neurotransmitters, the leftover neurotransmitters, are back in the presynaptic axon. And this cycle of reuptake and release repeats because we're recycling neurotransmitters. Now, there comes a time where neurotransmitters either degrade or we have what is called enzyme deactivation, where neurotransmitters are destroyed. And it's just one of the ways that the brain cleans up neurotransmitters. There's a limit to how many times we can recycle a neurotransmitter. So, with neurotransmitter reuptake, we are able to keep neurotransmitters in the synaptic gap longer. Stay with me here. So, our SSRIs actually cause reuptake. They block reuptake. And that is why, let me check my next slide. This is what helps speed up the brain when someone is depressed. The prefrontal cortex has become sluggish. We need to speed it back up to its normal state, and SSRIs and NSRIs, so serotonin, selective serotonin reuptake inhibitors, and norepinephrine and serotonin reuptake inhibitors, block recycling. That's all they do. So I think I have a better photo. So right here on this image, the green blocks here are actually blocking reuptake. So all of these neurotransmitters that have been released into the synaptic gap do not go back home. They don't go back to the axon terminal to be recycled. They stay in the synaptic gap. Now this is important when we use drugs that block reuptake. It's important because this allows, so using the example of SSRIs, this leaves serotonin in the synapse longer, making it more likely that serotonin will be absorbed by the postsynaptic neuron. Let me check my next slide. So when we come back here, if we block serotonin reuptake, I have all this leftover serotonin in the synaptic gap. Once my receptor sites reopen, they can absorb this leftover the remaining serotonin without my presynaptic neuron doing anything. So, as I explained, the prefrontal cortex becomes sluggish when we're depressed. This is what helps speed up the serotonin process. If I have blocked reuptake, my presynaptic neuron, the axon terminal on my presynaptic neuron, doesn't have to release serotonin in order for my postsynaptic neuron to receive neurotransmitters. And so as soon as these receptor sites open back up, serotonin can go into the receptor site. This speeds up the brain because now only part of the synapse has to engage for serotonin to be used. Okay. Now there are more than 30 to 100 neurotransmitters. I know, I wish they'd give us a more specific number than this, but that's what we've got. These neurotransmitters influence aspects of the mind and of behavior in humans. Now, other mammals are going to be using synaptic transmission too, but we're going to focus on human behavior and cognition. So, the emotions that you feel, the movements that you can make, your ability to sleep, to fall asleep, stay asleep, to dream, your ability to learn, to form memories, your ability to perceive pain and to control pain, anything your body does or your mind does is controlled by synaptic transmission. These neurotransmitters control what the brain is doing and when it's doing that. Now, when we look at neural communication, different neurotransmitters send different messages. So, as I mentioned, neurotransmitters are messages. They tell the postsynaptic neuron to either activate or stay at rest. And that is all neurons do. They activate or they stay at rest. So we talked about the cell body and the nucleus. This is where the message from the received neurotransmitters is processed so that the neuron can figure out, am I supposed to activate or am I supposed to stay at rest? So, when the presynaptic neuron, the axon terminal, sends neurotransmitters into the postsynaptic neuron, received by the dendrite, these neurotransmitters tell the postsynaptic neuron to either activate, and then it will engage in synaptic transmission, or to stay at rest. And so the neuron just stays in its resting potential. And I'll talk about that. Now, different neurotransmitters send different messages. So dopamine is an excitatory neurotransmitter. Let's see, GABA is an inhibitory neurotransmitter. So I want to look, I'll talk more about individual neurotransmitters in a little bit. Now, drugs and toxins can alter neurotransmitter actions. So there are what are called agonists and antagonists. An agonist enhances the actions of neurotransmitters. An antagonist inhibits the actions of neurotransmitters. So basically, an agonist is increasing the loudness of the message, where antagonists are making the message quieter, much more difficult to hear. Now, researchers can inject agonists and antagonists into animal brains as they're doing research to assess how neurotransmitters affect behavior. So here we've got an agonist, here we've got an antagonist. Agonist drugs increase how much of a neurotransmitter is created, so there is more of that neurotransmitter to release into the synapse, synaptic gap. They can also block reuptake, like an SSRI. Then there are more neurotransmitters available to receptor sites. So when reuptake is blocked, our postsynaptic neuron, these receptor sites are ready to go, ready to accept more of the neurotransmitter. So it keeps the message going, even if my presynaptic neuron isn't sending neurotransmitters. Antagonists decrease the amount of neurotransmitter. So there's less of there are fewer neurotransmitters within each synaptic vesicle. And we can create drugs that actually attack and destroy neurotransmitters in the synaptic gap. So antagonists can cause axon terminals to contain fewer neurotransmitters, and we can also create drugs that attack neurotransmitters in the synapse. Is much less likely to get to the postsynaptic. So let's look at a couple of